Hi, in this video we are going to talk about PCR. You can find written notes on what we are going to discuss today at medicinecanbeeasy.com. The link is also in the description. So what are you going to learn in this video? I am going to explain what PCR is, how it works and why it is so useful. But we are also going to discuss the limitations of the technique. Okay, so let's start. PCR, or polymerase chain reaction, is a very powerful laboratory technique that is used for copying segments of DNA. We already talked about copying DNA fragments in the previous video on cloning vectors, but as you will see, PCR is much more powerful. So first, what do we need to make PCR happen? Well, it is not a surprise that we need an enzyme that is going to do all the copying, and that is of course going to be DNA polymerase, which you should already know from DNA replication, for example. DNA polymerase needs primers to work properly, and because we are going to copy two DNA strands, we need one primer that is going to be complementary to the 5' end of one strand, and another one that is going to be complementary to the 3' end of the other strand. We also need 4 deoxyribonucleoside triphosphates, DNTPs, which the DNA polymerase will use as building blocks for the newly synthesized strands. We of course need the DNA we want to replicate, and finally we need a buffer, a special solution which contains magnesium ions and other salts that will create a suitable environment for the reaction. Now that we have everything ready, let's take a look at the process in detail. A typical PCR consists of 3 reactions, or we can say a chain of 3 reactions that are repeated in multiple cycles. For that reason, the technique is called polymerase chain reaction. The first step in the reaction is to separate the 2 strands using a process called denaturation. We simply heat the DNA to about 90 degrees Celsius, which causes the 2 strands to separate. Once the 2 strands are separated, we can proceed to the second step, which is called annealing. During this step, the temperature is lowered to about 40 to 65 degrees Celsius, and the primers attach or anneal to the 2 DNA strands, more specifically to the target DNA sequence we want to amplify, we want to copy. In the final step called extension, we again heat the mixture to about 70 degrees Celsius, and DNA polymerase synthesizes the new DNA strands in the 5' to 3' direction. At the end of the cycle, we get 2 double-stranded DNA molecules with the DNA sequence we wanted to copy. Each cycle of these 3 reactions only takes a few minutes, so you can imagine how much DNA can be copied in an hour if after the second cycle we already have 4 DNA products. Before we move on, we should also mention 2 important things. First, because the temperatures in PCR change rapidly, we need a special kind of DNA polymerase that is stable at high temperatures. For that reason, we use special polymerase called Taq polymerase, which was originally isolated from a bacterium that lives in hot springs. The second thing is that we use special machines for the reaction, which are called thermocyclers, and which are capable of changing the temperatures that we need for the reactions very quickly. So now that we described how PCR works, let's discuss how we can actually use the technique. PCR can amplify even small amounts of DNA, and for that reason is very useful in forensics. As we saw during the COVID-19 pandemic, PCR can also be used to detect the presence of a virus. If the primers complementary to the viral DNA anneal and the viral DNA is replicated, the presence of the virus is confirmed. PCR can also be used in medical genetics for screening mutations. Of course, these are not all the applications of PCR, but just something to give you an idea how useful the technique is. You should, however, realize that even though PCR is a powerful method, it has its limitations. Perhaps you already realized the first one, which is that we need to know the nucleotide sequence of the DNA segment we want to amplify so that we can construct complementary primers. We also said that PCR can copy even small amounts of DNA, and for that reason contamination in the laboratory can present a serious problem. Finally, PCR can only amplify short DNA sequences and is therefore not a suitable technique for amplifying large nucleotide sequences. Okay? So this is it. Thank you so much for watching. If you found this video helpful and you'd like to watch more videos on genetics or medicine in general, please subscribe. Thank you again and see you soon.